Hello, everyone. Welcome to today's HMSC research seminar. This is our second hybrid seminar, and we're asking everyone online to mute themselves, and please in here, if you mute yourself too. But seriously, if you have a cell phone, could you just mute your cell phone or put it on vibrate? No private chats, and we're going to hide all the non-video participants. All right, so welcome. Thank you for all joining us today for the HMSC Research Seminar. My name is Yi-Chung Chung, and I am the Academic Program Manager at Oregon State University's Hatfield Marine Science Center here in Oregon, Newport. And I'll be your host for today's talk. We also have Michael Banks here, who is the instructor for those of you taking seminar for credit. For those online, Sana Banks is our hybrid support today. Please let her know if you're having any technical issues so she can assist or let those of us on site know. So just a couple of reminders and logistics. This fall seminar will be offered in two formats. Again, fully remote where everyone's online or hybrid. Today's event is, again, a hybrid with audience members here in our new Marine Studies building auditorium and at HMSC and online. At this time, the option to be on site is limited to those that work, study, or live at HMSC. Watch the seminar announcement each week to see the format. For those online, we have your mics, cameras, and screen shared turned off, and we ask that you save your questions for the end, and you can post your questions in the chat box found at the bottom or top of your screen at any time, and we'll answer questions at the end of this talk. If you put a question in chat, Michael will read it out for everyone. You're also welcome to raise your digital hand, and you'll be invited to unmute when it's time for you to ask the question. Sana will be letting you know when it's your turn and will help to unmute your mic. Now, for those of you in the room, face covers are required at all times. Please step into the designated eating area if you need food or drink. If you're not already, please make sure you sign in. Sign-in sheets right there. This is a requirement for on-site events for contact tracing. Restrooms are available in the hallway directly behind the auditorium. And again, we'll be handling questions at the end. If you have a question, please raise your hand and I'll bring this mic to you and you can ask the question through the mic so that folks online can hear. If you feel the ground shaking, please proceed to the roof of this building by the external ramp, the stairwells, or the elevator on the east side of the building. We will be recording this event and it will be posted in a few days on the HMSC website's past seminar page that Sana will post in the chat. Any questions before we move on?
Related to your comment about getting into an elevator. Are you serious?
Well, yeah, because that elevator goes up to the top and that is our evacuation route.
I understand that. My concern is with the electrical elevator.
Yes, and that specific elevator is specifically made and designed to function in the event of an earthquake. Yes. Very, very good point to make.
Especially
Thank
today.
you. Also, a couple announcements. Next week's seminar speaker on October 28th will be a hybrid event with on-site option for those at HMSC. Jessica Watson and Greg Kurtzakowski with the Oregon Department of Fish and Wildlife's Marine Resources Program will be giving the second talk in our Sustainable Fisheries mini-series titled Come Get Schooled, the policy and science behind the management of Oregon's highly migratory and coastal pelagic schooling species. There are more links for upcoming events on our HMSC website. I also am going to make a couple announcements. The Oregon Coast STEM Hub is seeking scientists and graduate students like all of you to work with K-12 teachers in the Oregon Marine Scientists and Educator Alliance, ORSE program. It connects math and science educators with scientists to create and pilot lessons centered around marine-focused anchoring phenomena. Application deadline is October 25th, and that link will be posted in the chat, and there are funds to go with that to support that collaboration. Application workshops for the Oregon Sea Grants 2022 graduate fellowships and scholarships is November 3rd. I would encourage you to check that out, and that will be posted also in the chat. Registration is now open for Oregon Sea Grants education program for fall for virtual family homeschool programming. Please check that out, and that link will also be in the chat. And then finally, these last two announcements, there is still time to register for the State of the Coast 2021 conference that happens virtually on October 28 and 29 next week. And then finally, the deadline for the Marine Studies Initiative Advancement Award is October 29th. And these awards are to advance and support cultivating transdisciplinary collaborations, including through focus on human dimensions of the oceans and coasts and other expanded areas. Those are will be posted in the chat too. And then finally, for today, Cynthia Moffitt, the research program manager for HMSC, is going to introduce today's speaker.
Hi, everyone. Can you see me and hear me okay?
Can't hear you. We can't hear you here. I'm going to bring it up. And let's try
Can you hear me now?
Yeah.
Does that sound okay, Chung?
I think so. And we also have a blank projector, so I'm going to put that up.
Well, everybody online and in the room, thank you so much for joining us today. And thank you for working through these challenges. I wish I could be there for this talk specifically. So today's talk is the first of a three-part series about the past, present, and future of Yaquina Bay. This series was inspired by the Department of Land Conservation and Development getting a grant to help update the Yaquina Bay Management Plan. This is a space use management plan that was created in the early 80s to meet statewide planning goal 16. But at that time, uses of the bay were very different. The physical and biological parameters were different. So in light of this update of this plan, a group of individuals, Mike Greabn, Ted DeWitt, Walt Nelson, and Bob Cowan, invited a few speakers to help us understand the current status of Yaquina Bay. Our first speaker is Jim Kaldy, who's in front of you there. Jim is an ecologist with the US EPA's Pacific Ecology Systems Division with the Pacific Coastal Ecology Branch located there in Hatfield. He's been in that position since 2001. He has a bachelor's degree in marine science from Long Island University, his master's in plant biology from the University of New Hampshire, and his PhD from the University of Texas. Jim does research in estuary ecology and biochemistry, and he's going to talk to us today about the past, present, and future of Zostera in Yaquina Bay and other Oregon estuaries. So Jim, I'm hoping the floor is yours and you can take it away. Thanks, everyone.
Thank you, Cinnamon.
Here we go. Yeah. All right. Thank you, Cinnamon, and everyone for the invitation. Thank you all for coming. It's nice to see folks in person, and this facility is awesome. This is a great lecture hall, so I'm really happy to have the opportunity to present to you today. So one of the things I'd like to talk a little bit about is that PCEB, Pacific Coastal Ecology Branch, has been here for a long time, and we've done a lot of different work in estuaries and marine systems for a long time. So my name's on the first here on the slide, but there's a lot of other folks that had a lot to do with the work that I'm presenting. The other thing that I'm going to tell you is there's not a lot of data in this presentation. There's a lot of photographs and it's based on observations that I've made working in the system for about two decades. So as I mentioned, there's a bunch of folks from PCEB shown here. Walt Nelson on the left was our branch chief for about 15 years and had a lot to do with guiding our research towards understanding what's going on in Oregon systems. And there's been a number of folks and publications, reports, and that form the basis of a lot of what I'm going to be talking about today. So I really, this isn't just me. This is a lot of work from a lot of different folks. So just getting into the presentation a little bit, what we're going to talk about is Seagrass 101. What are seagrasses? Why do we care? Talk a little bit about the plants that occur here in the Pacific Northwest, specifically Zoster marina and Zoster japonica. Look at some drivers of change and some ideas, or what I think are important research needs for this area. So Seagrass 101, what are these things? Seagrasses are flowering plants. They are monocots in the order Alismatales, and they're a fairly old group that has evolved from land plants that returned back to the ocean, to the sea. They have true roots, stems, and leaves, unlike algae. And they have xylem and phloem, so they do transport water and sugar using these highly developed systems. They're very well adapted to the marine environment. They're obligate halophytes. They need that salt water. They have hydrophilous pollination, and that's just a fancy way of saying that the water moves the pollen around. And they have flexible strap-like leaves to deal with the water currents and wave motion. And they also have this really neat system called a continuous parenchyma, or a lacunae system. And basically this is these air holes or tubes that extend from the very tip of the leaves all the way down to the tip of the roots, and allows the plants to live in very anoxic sediments. They can transport photosynthetically derived oxygen into their roots and support aerobic respiration. As I mentioned, they also, I don't know if you can see the cursor. Oh, you can, great. These little dots here, those are pollen grains that are transported between the released from the male flowers and fertilize the female flowers to form seeds. So that's sort of what they are. Why do we really care about them? And a lot of that has to do with ecosystem services. These plants take nitrogen out of the water column. They also take carbon out of the water, and that can lead to carbon sequestration. So they can add that carbon into the sediments, and that gets buried and is removed from the system. But they also provide a lot of habitat for various different types of animals, whether that's eggs for, or a place for herring to spawn. Salmonids live and spend part of their life history in the seagrass beds. All sorts of birds feed either directly on the plants or on the critters that live within the plant communities. And it's a great place for fish and crab to find refuge and food. So we have a few different species of seagrasses here in the Pacific Northwest. Is there a way to get rid of that bar, Yichung? I'm sorry? Okay, just grab it and, okay, let's try something like that. So there's a few different species here, and the two species of Zostera, Rupia marittima, and then 3 different species of Phyllospatix, which occur here. Phyllospatix is almost exclusively on the open coast. And the 2 that we're going to really focus on, okay, let's try it. Seem to have locked up here. There. I'm sorry. The 2 we're going to focus on are Zoster marina and Zoster japonica. So this is a photograph on the left showing me before I got gray, holding a Zoster marina plant collected out at the near the OSU Cove. These plants tend to be about 30 to 120 centimeters in length. I've seen them grow up to 3 meters in some places. The plants are fairly wide at about 12 millimeters, and there's a reasonable density, 60 to 140 shoots per meter squared, and biomass ranges around 30 to 200 grams dry weight. Japonica, on the other hand, is very different in the fact that it's very small, 10 to 20 centimeters. Sometimes we'll get up to 30 centimeters. Leaf widths are on the order of about 1 millimeter, and shoot densities are crazy, between 2,000 and 10,000 shoots per meter squared. Comparable biomass characteristics. And the other thing that's a bit different about them is that Japonica tends to live very high in the upper intertidal, around +1 to +2.5 meters above mean low or low water. So let's try that. I wanted to just briefly talk about some of the physiological requirements. These plants have very high light requirements, so they need to have clear water. And the reason for this is that they have a large amount of non-photosynthetic biomass that occurs in the sediments. And so they have to have a lot of light to photosynthesize to support that tissue that lives below ground. They're also very sensitive to temperature. Zostera marina has an optimal temperature for growth and photosynthesis between 15 and 20 degrees C. Higher than 25 degrees C, it is very stressed, and greater than 30 degrees C, those plants will die quickly. Japonica, on the other hand, likes the water a little bit warmer, around 20 degrees C, and 32 to 35 can be lethal. They have a very wide salinity tolerance. And nutrients in the Pacific Northwest, the water column nutrients are so high that these plants are pretty well nitrogen replete. But temperature definitely seems to have a bigger impact than nutrients. And we'll talk quite a bit more about this a little bit later on. So to show you some of this, the panel on the left here shows net primary productivity versus temperature. This is a panel from Ron Thom, some of his research. And you can see the highest point here is around 15 degrees C. And as you get warmer, the net primary productivity decreases. And this is in response to the fact that the respiration rate increases much more quickly at warm temperatures. So as the temperature increases, respiration increases faster than photosynthesis. And as a result, you can end up with this in a negative carbon balance sort of situation. I'm going to try moving this again. Let's cover EPA because that's the same on all of them. All right. Okay. It doesn't like it when I move that thing on the slide. There we go. So this is a photograph of eelgrass, Zostera marina, taken underwater here at OSU Cove. Eelgrass is the common name for this plant, and it was originally described by Linnaeus. So I'm going to talk a little bit about locations of some large-scale Zostera marina losses. We're going to look at places like Westcott Bay, Coos Bay, and Morro Bay, as well as a few other locations. The map on the far right there just shows the distribution, the dots there show the distribution of Zostera marina along the West Coast. So this is Westcott Bay in the San Juan Islands. And between 2001 and 2003, they lost about 20 hectares of eelgrass. It just sort of disappeared almost overnight. Never really been able to identify a direct cause of what happened. And restoration efforts have not been successful in this particular area. And you can see the orange highlighted areas are where eelgrass is essentially gone from now. It used to occur there, but it's gone. And then there are some other places over here in the green that seagrass continues to thrive. In Morro Bay, California, there had been some consistent mapping since about 2002. Earlier efforts were inconsistent, but they did have some data about that. And estimates were on the order of about 400 acres of seagrass. From 95 to 2010, there were variable estimates. Some of that was based on methods. And then between 2007 and 2016, there was a 90% decline in eelgrass. And this is sort of shown on the panels on the right-hand side of the screen. The red indicates where eelgrass occurred, and then you can see that it's very disappears. By 2013, there was only 15 acres of seagrass left. And they're starting to see some evidence of colonization of natural colonization reoccurring, but restoration efforts started in 2019, but they have not been all that successful. So in South Slough, a little bit closer to home, there's been another documented loss of eelgrass from at Volino Island in particular. Annual monitoring has been going on there since 2004. There've been quarterly sampling as part of the Seagrass Net, which is a network of seagrass monitoring stations around the world. And prior to 2016, the Volino Island population had been very stable for about 11 years. In 2016, they noted a decrease. And then between 2016 and 2019, there was a 50% loss of eelgrass. And this is shown on the, you can see in the photographs, 2002 to 2018, big differences, roughly the same areas. And you can see here, this is mapping work done by a master's student, Anderson 2020. The green is seagrass, and then in 2019, there's only about 50% of that is left, and it's much sparser and more disjunct. Puget Sound, the Washington State Department of Natural Resources does an amazing job of monitoring seagrass beds throughout the entire Puget Sound. This is some data and text taken from their 2016-2017 report that came out in 2019. And you can see that most of the areas where they're seeing loss tend to be sort of these central Puget Sound areas in here, and then down in like places like Hood Canal at the end of these deep fjords that don't get a lot of water movement. And they also noted that there's that these losses seem to be related to unusual environmental conditions. Up at Sequim, Washington, colleagues Ron Thom and Amy Bordy have noticed some seagrass loss up in this area, roughly 700 square meters. Here you can see the red area that's been lost. This occurred during the blob event, marine heat wave a few years ago. And there were similar trends at other Washington DNR sites. One of the interesting things is they also noted the presence of Canada geese, and we'll talk about that a little bit later. So what do these losses of seagrass at other bays have to do with Yaquina? You know, this is a photograph. This is actually a digital photograph of a print photograph from 2002 when I first started working here. This photograph shows a transect through the eelgrass bed directly across from the LNG tank. You can see here, it's a 50-meter transect. The little white flags are 10 meters apart. And so the transect starts right at the water line, which on this day was a 0-0 tide. So pretty much mean low or low water. And you can see it's a fairly wide seagrass bed, at least 50 meters. So a friend of mine said, this is Fiona Tomas Nash, had driven across the Yaquina Bay Bridge and said, no, the seagrass looks like it's really not doing as well as it used to. And so I got a wild bug across my butt to go out and take a look at some of these historical sites. And this is what I found at one of my old research sites on Yaquina Bay. You can see the bridge in the background, HMSC back here. This was a minus almost 2-foot tide in April. And the water, the deep edge of the bed is out here about where the change in the water waves are. And then the upper edge is here. But the bed to me looked much narrower than what I remembered and compared to the photographs like that. So Again, this is just sort of anecdotal, am I remembering these things correctly, or something changed, or what? So I did a quick GPS study, took a logging GPS and walked the upper margin of the seagrass bed. I believe this was in early June here in Yaquina Bay. I started up here by the seawater intake and walked almost all the way down to the King Slough entrance. And that pink line represents the GPS track that I walked right along the upper edge of the seagrass. And that's overlaid on a high resolution aerial photography classification of seagrass from 2007 that was done by Pat Clinton and David Young. So that was ground truth seagrass data, a map of eelgrass within Yaquina Bay. And that pink line is pretty radically different from where the upper margin of eelgrass used to be. You can see that here in this inset here. It looks like 40 to 50% of the eelgrass bed seems to have disappeared. The yellow dots there and the blue dots represent some places where we had very high, good quality GPS data on the upper margins of the eelgrass, upper and lower margins of the eelgrass bed, again from 2002 and 2005. And it really appears that there's been some big changes to Zoster Marina here locally. So we're going to switch gears a little bit and talk about Zoster Japonica. This is a non-native plant. It's also known as dwarf eelgrass or Japanese eelgrass. And this, again, is an underwater photograph from OSU Cove from a few years ago. So Japonica was introduced to Washington State. It's believed to have come over with oyster aquaculture in the early parts of the 1900s. The oyster spat and oysters were packed with Japonica leaves, plants to maintain moisture during transit from Japan to the US. And the first noted collection of rooted Zoster Japonica was from 1957 in Willapa Bay. And then it sort of spread throughout the region. In 1974 and 75, it was found in Puget Sound, Boundary Bay. 76, Range Bear, who was worked here for decades, had documented it here in Yaquina Bay. In 1978, Fraser River, 79, Vancouver Island. In the early 2000, Scott Larned, who was a postdoc at the PCEB lab, did an informal survey of estuaries along the Oregon coast and found Japonica in 60% of those bays. In 2002, Japonica was documented in Humboldt Bay down in California. And then in 2012, Washington State had declared Japonica to be a noxious weed. So we'll carry on from there. This is some of the mapping work that David Young and Pat Clinton and David Specht had conducted over the years. And in 1999, they had done, again, aerial mapping surveys and noted that Japonica was just a little bit, oops, there we go, oh darn. It's just a little bit of Japonica here in Sally's Bend, tiny bit here on Idaho Point, and not much anywhere else. By 2007, Sally's Bend was pretty well infested. Occurs here in King Slough, also here on Idaho Point. And it had expanded from about 3.7 hectares in 1998 to about 19 hectares in 2007. In comparison, Zoster Marina covers about 1 square kilometer within the estuary. So it wasn't a huge amount of eelgrass, but it was a huge proportional increase. Looking at the Japonica in a little bit more detail here on Idaho Point, The black dots in this figure represent Japonica in 2000, 0.09 hectares, and the white indicates in 2014 what was present, and it was 0.57 hectares. So again, this big increase in very localized areas. So what does these places look like today? So this is a photograph that I took in 2002 in Zoster Japonica bed, where I had been doing research. In 2009, you can see the Japonica is still present, a little bit different distribution, not nearly as dense. But then in 2021, just a few months ago, I was out there and there was almost no Zoster Japonica there at all. So this is the same tree in these three photographs. And the green that you see back here is macroalgae. It's not Japonica. There are a few patches of Japonica, but not nearly what it used to look like. So these are the same areas here outlined in yellow. There's a little patch in there, and this is what it looks like a little bit closer up, and that's about 6 feet long. So pretty big change in the Japonica distribution at this particular site. There's a number of other places that it has expanded, and a number of other places that it has just sort of winked out. In OSU Cove, there's almost none left, and it used to be a pretty good sized bed there. So looking at Sally's Bend, 2011, I had done a bunch of work with Deb Schaefer, who worked with Army Corps, since retired. Her and I had collected extensively here on Sally's Bend, and this is her boots that got left in the mud that day for a little bit. But you can see how dense the plants are, and this is just extensive throughout the whole area. Looked like this. This summer in 2021, same area. The plants are still there. There's Japonica present, but the density is radically different, or appears to be radically different. I don't have quantitative estimates of that, but visually they're quite striking. Humboldt Bay was a really interesting situation. As I said, it was first discovered in 2002. They undertook a number of management actions to try to eradicate Japonica in Humboldt, and they were successful here at the Indian Islands sites. They were able to get rid of most of the Japonica. Other sites were not so successful, and there were changes in the number of patches and the patch area, and we'll talk about that in a few seconds. But there had been Japonica documented here at Manila, Wallace Ranch, and all of these sites over here. So at the Manila site in 2009, 2010, there was a 50% increase of 3 patches and a 6-fold increase in the patch size. So they got 6 times bigger. At the Arcata Marsh in 2010, there were 130 patches that were accounted for over 2,000 square meters. A 10-fold increase in patch number and a 20-fold increase in patch size at Arcata Marsh, 4-fold increase in number and 9-fold increase in size at the wastewater treatment plant. It was also documented in the Eel River estuary, very dense. This is part of a photograph from Eel River, a very dense bed here along the upper edge. In Northern California. They did some experimental work in this area trying to eradicate, and they're working at small patch size, maybe half to 1 meter square areas, and they were not successful. Unfortunately, the monitoring program ran out of money, and they basically went away and started, ignoring the problem. In 2017, I had the opportunity to go and sample with some colleagues from China to collect genetic material samples. And Cal Fish and Game was very helpful. They sent people out to reconnoiter and find some Japonica for us. And they went to the Eel River and it was gone. This bed no longer exists. Nobody knows why. It wasn't a result of the eradication efforts, but it disappeared. So this is the group of Chinese colleagues that we sampled Japonica up and down the West Coast. At the Manila site, this was the one patch of seagrass that we found. You can see it's really sparse. That's maybe a meter in diameter patch. And then at the Arcata wastewater treatment plant, we didn't find any. That's just completely bare mud habitat. There was no Japonica there at all. So what's going on? You know, we're seeing these large scale declines and loss of the native eelgrass, Zoster marina, up and down the West Coast. We're seeing some changes locally. We're seeing local changes in Zoster Japonica, and that may also be happening at a larger scale. But there's not really a lot of good data and evidence on that. What causes the declines of seagrass? This photograph on the right is, my right, your left, is a photograph of South Beach, this stretch of beach that I walk every day with my dogs. And at certain times of the year, you'll see just big piles of this eelgrass rack that wash up coming out of the bays. So what's going on? So there are a bunch of things that cause declines in eelgrass. I mentioned, you know, we talked about light earlier. Light's big. These plants are very sensitive to low light conditions, but there really doesn't seem to be much changes in water clarity here. Wasting disease can be a big factor. Labyrinthial Zostera is a marine slime mold that has been implicated in the loss of Zoster marina on the East Coast and in Europe in the 1930s. Eelgrass was almost eliminated from most bays and estuaries on the Atlantic. It also hit the West Coast, but not as hard. You know, so it definitely can have an impact. Avian overgrazing can cause loss of eelgrass. That's been documented in New Hampshire. They saw some evidence for that up at Squim Lab and at PNNL. When I was out earlier this summer, I took this photograph of the Canada geese chewing on the meristems, and then, you know, you walk by and find floating plants. So they definitely can have an impact. But I'm not sure that that's what's going on. We also have the temperature stressors. You know, there's these long-term kind of chronic increases in water temperature that are occurring, but we're also getting these episodic warming events, things like the marine heat waves and ENSO events, changing the thermal characteristics of the water masses that are sticking around. So this is the blob year 2014, and, you know, this year in September of 2020. In August of 2021, there was a prediction that there's going to be another marine heat wave hitting potentially hitting Oregon, Washington, California. I stopped paying attention, so I'm not sure if that's actually happening now or not, but this was predicted to be occurring about a month ago. So this plot that came from a NOAA page, and it's showing the area of marine heat waves, the total surface area of heat waves. And I'm using that for a proxy of when these things occurred. You know, I mentioned, well, maybe I didn't. Willapa Bay saw some decreases in chute density. This is some work done by Ron Thom in the early 2000s and with pincers program. They looked at chute density in a variety of different places, including Willapa and I believe Coos, and they related changes in plant density to ENSO events, El Nino, La Nina type things, where the warm water coming in was causing chute density declines. You know, and so I plotted some of these the time frame of some of these other times that we've seen seagrass losses, or roughly the time frames, and they seem to be pretty well correlated, particularly the more recent ones here with these large scale marine heat wave events. I think there's a correlation there. Again, I said that there's not going to be a lot of data here. This is me just looking at it going, looks like there's something happening here. So have we seen this sort of thing in other places? And the answer is yes. Shark Bay is a World Heritage Site in Australia, and they were hit with a very strong marine heat wave in 2010 and 2011, lasted for about 8 weeks, or roughly 2 months. And water temperatures were up to 4 degrees C warmer than normal. And you can see sort of the impact of this on the 2 panels here, 2002 and 2014. The dense seagrass shown in the dark gray. In 2002, there's a lot more area than there was in 2014. This plot, just the red here, shows the actual seagrass loss a little bit better for folks. I'm red-green colorblind, so sometimes this stuff doesn't always work out great for me, but for a lot of other people, to see that red just sort of drives it home. They lost 1,300 kilometers of seagrass. If you remember, Yaquina Bay has about 1 square kilometer of seagrass, so it tells you how big that system is. And they were looking at mostly Amphibolus and Placidonia, so different species than what we see. One of the things I found really interesting was that the shoot death actually occurred 1 to 2 years after the marine heat wave event. And that sort of lines up with some of the things that we were seeing on that previous figure, that it seemed like there was a big marine heat wave, and then it was sort of a lag of a year or 2 before there seemed to be a noticeable decline in seagrass. There've been documentation of extensive eelgrass loss on both the east coast of North America and in Europe. So this is my eelgrass mechanistic response cartoon to water temperature, how these plants respond to water temperature. Under optimum temperatures, the plants are really happy. They've got, oh, those came out funny. The emojis don't look quite right. Oh, well. There's a lot of photosynthesis, a little bit of respiration. So they've got a strong net primary productivity, lots of carbohydrate being stored. They're happy plants. At sort of this moderate warming, they've got still good photosynthesis occurring. Respiration is increasing. So there's a little bit of a decline in net primary productivity. Carbohydrates are still being stored, but not quite as much. And the plants are doing sort of meh. They're doing okay. As the temperatures continue to warm a bit, photosynthesis occurs, but respiration is really becoming much more dominant. There's a loss, negative carbon balance, so net primary productivity is decreasing strongly. Carbohydrate reserves are being impacted. The plants are not doing well. If temperature increases continue, it can lead to plant death. The other thing, once these plants get stressed and they're unhappy, other stressors, things like grazing or wasting disease or light limitation can really take a toll, further exacerbating this, the plant's not doing very well and ultimately leading to shoot death. So that's sort of my cartoon of what I think is going on here. What I see as some future research needs, I really think there's a need to update the maps of Zoster Marina and Japonica within Yaquina Bay. I'd expand that to the whole coast of Oregon and Washington, California. I think seagrass mapping can really go a long way and cover some new ground. And it hasn't happened in quite a while. I think it would be great to initiate and maintain a seagrass monitoring site, something like Seagrass Net. We'll talk a little bit more about that here in a moment. Just to sort of set that baseline and provide that idea, that canary in the coal mine of how what is the health of the ecosystem as you would determine from the plants. And continued research on multiple stressors and how those stressors interact to potentially stress out the plants and lead to, if that leads to declines or whether or not they can tolerate it, what are those threshold limits. And I think there really ought to be a prioritization of conservation efforts over restoration. Seagrass restoration is notoriously poor success rate. It's very difficult to do well. I think conservation should be maximized. So I mentioned Seagrass Net a couple of times. This was originally developed by Fred Short at UNH, University of New Hampshire, and there's 112 sites currently in 30 different countries. It's recently been turned over to the Smithsonian Institution. They've taken over the project and are working to bring everything back online. But it's a very well documented monitoring protocol. Would be ideal for citizen science or recurring class, project sort of thing. The methods are all worked out. There's a quality assurance project plan. They've got the data sheets. It's a very well designed monitoring program, and it could provide a lot of useful information. As I said at the beginning of the talk, there was a lot of folks that participated behind the scenes. My name was on the first slide there, but I couldn't have pulled all of this together without discussions and help and just colleagues bouncing ideas off of colleagues, whether they've been at PCEB or Hatfield or other institutions. And Nate Lewis was particularly helpful with plotting that GPS data. And I'd like to leave you with just some interesting photographs that I've taken over the years. The snow in South Beach, the elk on the marsh here in Yaquina kind of blew my mind. The mud flat with a full moon, going out to work on the mud flat, and some of the amazing sunsets that we have here. And that's my presentation. So if anybody has questions, be happy to give it a go. Thank you.
Do we have any questions from the audience? Online? Do we have questions online?
Gail Hansen, does increased sedimentation influence the populations?
It certainly could. The sedimentation would obviously, there would be sediment suspended in the water column, and that would cut down on light and definitely could be a negative impact to plants. One of the things that's kind of interesting, when we look at sort of the water quality data, things like the sondes that they have at South Slough, but also here at Yaquina at the water intake, you don't see a lot of turbidity. There's not a lot of suspended solids. There's episodic events, but nothing sustained. And these plants can really tolerate weeks at very low light levels. And there was some work that I had done earlier in the 2000s where we found that the plants here in Yaquina Bay could survive for a couple of weeks with very, very low light levels, below 3 moles per meter squared per day for long periods of time, because they got a little bit of light and they're able to photosynthesize a little bit. But yeah, I mean, that definitely could be an impact. But I think there's some other there's some other things that we need to consider as well.
Hi, can you hear me, Jim?
Yes.
Hey, Jim, thank you for sharing your work. I want to ask about the question about the nursery habitat function. We think about seagrass as really important. That's kind of the conceptual model. Really important nursery habitats for crabs, rockfish, you name it. Given the fluctuations, if not declines, have people picked up a signal of the impact of habitat loss? Are we ready? Are we even ready to detect such a signal?
Oh, that's a really good question. And I don't think I have a really good answer for you. You know, a lot of the monitoring programs are currently pretty coarse. And, you know, as I was talking about, the highly, you know, real detailed maps that Pat and David produced, that was a lot of effort to develop those maps. I don't think that there's necessarily been the corresponding work to tie together, you know, changes in seagrass extent to changes in animal populations. I'm at least, I mean, that work may be out there, but I'm not aware of it. So I think that's a really interesting question. And it's one that's kind of crossed my mind is, okay, you know, there appears to have been this change in the grass beds. You know, I'd like to see some more definitive data on that as well. But you know, how does that trickle down to the other trophic levels that rely on the seagrass? And I don't have a good answer for that, Francis. Thank you for asking the question, though.
Yeah, from David Thompson. Science published a study that found eelgrass was genetically more robust when sea otters were actively disturbing them. Climate change is generally too rapid for evolution to respond to it, but without otters facilitating more sexual crosses, might they be more further jeopardized?
Ooh, wow, that's an interesting question too. Yeah, I know that this is one of the things that I would kind of allude to on that stressor research. There has been a fair amount of work showing that different genetic strains of eelgrass respond to stressors differently. And I've heard some talk about, you know, trying to select and restore seagrass beds using plants that are more resistant or resilient to warmer temperatures and things like that. That's probably just in its infancy, and I'm not aware of much more than that. But I think that's a really interesting question there to look at how the genetics of populations influences the community response to these bigger stressors. I think that's a great field of research for folks to look at. But I don't know much more than that.
I'll go, and then there you go.
I was wondering, since the ecosystem services that eelgrass is providing is a lot of sheltering, I was wondering if different patch sizes and shapes, if you looked at different like uses, and if the size and shape has a big effect.
So folks have done a fair amount of that work, not necessarily with eelgrass per se. I'm more aware of work like that with Thalassia and Haliduli in the Gulf of Mexico, where they have done a fair amount of work looking at the relationship between patch size and the ability of prey items to hide from predators, and basically looking along transects to see if there are differences in where the animals hang out. Are they hanging out at the edges or the center of the patches and that sort of thing? So there has been work done on that. How that fits to Zoster Marina, I'm not exactly sure. But yeah, there is a lot of interest, research interest in that sort of thing.
And from Ryan, you mentioned eradication efforts. Why are certain areas looking to eradicate seagrass if it's so important to local ecosystems?
Wow, that's a loaded question because it turns out there's a lot of politics involved. In some areas, the eradication has been driven by the aquaculture industry. There's a competition between Zoster Japonica and some of the Manila clam aquaculture. The distribution of the two different organisms overlap, and the aquaculture interests find it harder to harvest and a little bit lower yields of their target species. In other places, that's been more driven by the quote unquote invasive nature, in that, yes, there are ecosystem services, but this is a non-native plant. It doesn't belong here. Get it the heck out. And they've done all sorts of really interesting eradication studies, things using herbicides to flamethrowers to injecting hot water into the sediments, I mean, which kills not only the seagrass, but it kills anything else that's in the mud. So it's really been an interesting thing, but it kind of comes down to this society values and societal values of what is an ecosystem, what are the services that these plants provide, and does it outweigh do the benefits outweigh the negatives? And the answer is it depends on who you are and what your perspective is. Right on.
Yeah. Hey, I got in here a little late, so I apologize. But it's always been my impression that the Japonica kind of pushed out the native stuff, right? Have I got that right?
Actually, for the most part, in most places, they don't overlap.
Well, they don't overlap.
There are a few places in Willapa Bay and up in Padilla Bay that the two species do coexist. But here in Yaquina Bay, the Japonica is up in the real upper intertidal, about +2 meters. And most of the Zostera marina, the native plant, is down around the lower, around mean low water, so the 0 tide mark, roughly like -2 to about +1 foot above mean low water. So they're really disjunct in a lot of places. There are a few places where they do co-occur, but in most places they don't really interact all that much.
More resilient to these heat waves.
So I don't think so. That's my working pet hypothesis, is that these warming events and the warm temperatures are what's causing some of these seagrass declines. Unfortunately, again, I don't have the data to say that definitively one way or the other. But I keep thinking about the scale of these changes, from Southern California up to, into the Puget Sound, that we're seeing changes in these seagrass beds. And the only thing that's really occurring at that sort of large scale are these marine heat waves. And when you start thinking about the physiology of how the plants respond to how they respond to temperature, with the increase in respiration occurring much more quickly and the net primary productivity decreasing, I think that's a much more likely explanation. But again, there's all sorts of other possible explanations as well. It could be wasting disease. It could be changes in grazing pressure, or all of the above. But I think that there's a strong case to be made for the temperature issue. My pleasure.
Question from Maria Jose. How do seed banks play a part in these changes?
That is really, that's an excellent question. And I don't have a good answer. So Zoster Marina does have a little bit of a seed bank. They'll last for, you know, a season or two. And one of the things that, you know, but gosh, I don't have a good answer. I suspect that it does play more of a role in the resilience side of things, and that once a bed has been sort of knocked back, that hopefully more seedlings would come in and establish. But the typical sort of paradigm in seagrass ecology is that most of the growth of a patch or a bed is through clonal reproduction and not necessarily through genetic reproduction or through seeds. But when you start looking at the genetics of populations, there's a lot more diversity than you might expect if that were really the case. So I think there's a lot to be learned, a lot to be done in that to answer that question still.
We're nearing the end of our time, and I know we do have a few questions online as well as on site here. So I would encourage folks to ask your questions. Jim's email address is on the slide.
Please.
And for folks who are here, you're welcome to come and actually speak to Jim directly about any questions that you might have. Again, a round of applause. Thank you, Jim,
Thank
for
you.
coming and talking. And thank you everyone for joining us online and in person today. And the recorded talk will be on our website. Thank you.
Thanks.